a great afternoon session, and I just wanted to highlight the fact that a number of the board members of CRF are going to be involved in this with doing introductions, and our first intro board member is John Trott. So thanks, and we're really looking forward to the afternoon session.
All right, come on in. Okay, come on in. All right, so great. Thank you very much, Neil. All right, good. The mic works too. So yeah, it works fine. So it's my pleasure to start off the afternoon as we're talking about clinical trials in choroideremia, and it's my pleasure to intro our first speaker. Dr. Jess Thompson is the head of our or runs our research committee, and I go to those meetings all the time, and it's absolutely invaluable to have a physician actually doing this because a lot of us are winging it as we go and understanding the data being presented and the proposals and such. But Dr. Thompson is a pediatric thoracic surgeon. Recently now he moved in Arizona, so now he's in Prescott, Arizona. His wife is a carrier, he's an affected son, and so he's very much involved in in the overall Choroideremia Research Foundation, but an indispensable leader for our research committee and the tireless interface between the CRF and the research community. So, with that, I'd like to introduce Dr. Jess Thompson.
Hello, everyone. Good afternoon. My name is Jess Thompson, and it's a pleasure to be here. I'm grateful to the CRF for the opportunity to present a little bit of information. I'm not sure how to get this to stay, but I also noticed that I do have an error on this first slide. My email actually is not jess.thompson. It's just jessthompson@curechm.org. So please feel free to reach out to me. Oh, thanks. Thanks. Whether we'll have a little Q&A after this, or feel free to call me or email me with research questions, ideas, thoughts, or comments. Would really appreciate and value your input. For a few minutes today, I would just like to kind of give a brief overview about what the research committee does, who we are, what our purpose is, the strategic way that we use our funds to fund research, and then a summary of some of the funded projects that we have ongoing. So just to go back for a second, the purpose of the research committee for the Choroideremia Research Foundation, our overall objective is to search for a cure for choroideremia. So as such, the research committee, we review, rank, and submit to the board fundable research proposals. We also advise the Choroideremia Research Foundation and its members on research matters. We keep our organization abreast of state-of-the-art discoveries. We also administer the Scientific Advisory Board. And finally, we encourage and promote collaboration between the investigators, not just in North America, but globally. These are the members of the research committee: Neil Bench, Peter Boone, Yair Einhorn, Beth Foss, Nama Goldman, Ariel Harp, Eric Hartman, Robert Hillier, Sandra Hoffman, Corey McDonald, Chris Moen, Mike Mullen, J.R. Rizzo, John Trott, Laurent Parod, Mary Porter, Tracy Pretorius, myself, Jonathan Wells, and Mengling Jiang, and finally Kathy Wagner. In addition to the research committee, we have a scientific advisory board. I chair that board. We then have Dr. Alleman from University of Pennsylvania, Dr. Barty from the NIH, Dr. Duncan from UCSF, Dr. Kim Edwards, who is working on her PhD, and she's a recipient of the Wheelock Award 2 years ago, Dr. Huckfeldt from Harvard, Iana Kone from Duke, Dr. McCall from Louisville, Chris Moen, Mark Panizzi from Oregon, and then Cynthia Kwan, who Dr. Kwan also received last year's Wheelock Award, Dr. Sang from Columbia, Dr. Vincent from Sick Children's, and then Dr. Young from Harvard. So each month we have a research committee meeting. So those members that are on the research committee, we meet to review the current state of our grants, the current research that's out there, and then any grant proposals that have come in. Then every quarter the scientific advisory board meets as a whole, and the primary purpose of these meetings are twofold. Number one, it's to review and rank any grants that have come in, and number two, to have a bright and forthright discussion of in which direction is the field moving, so that we know where we need to focus our time, attention, and monies. And then as members of the research committee, we present to the general board on research matters and in terms of what projects we feel should be funded. And then we also meet as needed. So as you can see that the members of the research committee were a quite active group. As I mentioned earlier in the introduction, we have a very specific objectives when we want to fund a research proposal. Primarily, we are looking at investigators and the significance of that project that's been asked to be funded. In terms of investigators, one of our primary aims is to continue supporting investigators that have worked with us in the past and have been productive in the past. Also, one of our very strategic objectives is to find new investigators or emerging investigators. Our hope is that by supporting these investigators early in their career, that we can develop a relationship with them and that they can develop their career such that choroideremia is not just a side focus of their overall mission of their research projects, but it's the main mission of their research projects. The other goal that we have in terms of our funding projects is to look for projects that will allow for collaboration between established researchers with whom we've worked in the past, or perhaps linking an established researcher with a new or emerging researcher in the field of choroideremia. In addition to looking at the investigators themselves, we want to understand, or we only want to fund projects that have real significance. So we want to look for projects that are foundational to the discovery of therapeutics for choroideremia or the basic pathophysiology of choroideremia. And really, we're looking for projects that will advance the field forward. We are not looking for projects that are just one-offs. It's an interesting thought, an interesting idea, but then it has a dead end. What we do when we're looking for grants to fund, one of our first questions is, okay, so if this project, if the outcomes of this project are as anticipated, how will you use the results of this project to further your research, and what's the next project going to be? So again, over the past several years, we've tried to be very purposeful in our funding approach. One of the things that we've looked at heavily in the last several years are female carriers. Up until now, the overwhelming majority of research that's been done has been on the male carriers of choroideremia. And little to none has been done on the female carriers, and we're starting to rectify this by promoting several research projects that look specifically at female carriers. Another area of strategic interest that has come across lately has been the development of new animal models. We heard a little bit in this morning's session about a rodent model. That model we're trying to refine, and we're about to hear about a larger animal model in just a few minutes. Also, we heard in this morning's session about 3 therapeutic trials that had to deal with gene therapy and gene manipulation. And so we're always very keen to fund projects in that space, whether it's DNA, RNA, or even non-viral vectors. And then in addition to genetic therapeutics, we're always looking for alternative therapeutics, and because we might not be able to cure choroideremia, but maybe we could arrest it, or maybe we could recover people that have suffered the effects of choroideremia. And so these right now are the 4 central areas that we're funding projects in. Just to give you an overview of the projects that we funded for the last 5 years, in the last 5 years from 2018 to 2020, we funded a total of 27 projects. This year to date, in the first 6 months of 2022, we funded 4 projects. We funded 8 projects in 2020 and 8 projects in 2021. And those, we had done 2 projects in 2019 and 5 projects in 2018. So you can see that we're really trying to fund more projects here in the recent past. In addition to members providing us money, and it just goes into a general pool, we have had the various generous support of particular individuals or family foundations for named funding sources. I'll just spend a moment. Randy Wheelock, I met him once in person, had several conversations with him on the phone. This was an individual who made choroideremia research truly his mission in life, and unfortunately and untimely passed away a few years ago. And in his honor, the Choroideremia Research Foundation has made a research award. He actually came up with the idea for an award. He didn't say an award after me, but he came up with the idea of an award for new investigators. And so we have awarded this twice, and we're in the process of evaluating our current grants for the third Randy Wheelock Award. What's nice about the Wheelock Award is those young investigators that are being supported financially from the CRF, they are also invited to be on our science advisory board. So not only do they get an opportunity to pursue research in the choroideremia research space, but they also get to see and be mentored as to how a scientific advisory board works. And as you can see, what we're trying to do is nurture these people so that they can stay with us for the rest of their career. We've also had funding from the Bourne family, the Oster family, Salois family, the Auburn Theta Chi Chapter Award, and the Einhorn family. In addition to monies coming from from the United States, as you can see here, CRF Canada has funded us. We have co-funded with them 11 of the on almost basically half of the projects that we've had in the last 5 years. Bob Hillier, the president of Canada CRF, is with us today, and he's always very, very helpful, and that organization has been very helpful. And as you can see, we actually have a lot of funds that go to Canada. Fight for Sight has also helped us fund a project in the last 5 years, and the University of Pennsylvania Orphan Disease Center has also helped on 4 projects. And we have collaborated with them for several years now in raising funds for their organization through the $1 Million Bike Ride and other charitable events. These are the institutions that we have funded. Just because you're up here once doesn't mean it's only one grant. So for example, the University of Wisconsin, we funded them multiple times over the past several years, and we're going to hear from one of their PIs, primary investigators, Dr. David Gamm. We funded University of Pennsylvania, UCLA, University of Michigan, and Renovate Biosciences here in North America, and we're going to hear more about that in just a few minutes. Outside of the United States and Canada, we've given funds to the University of Alberta and University of Montreal, in England across the pond at Oxford and the University College London, in France, Montpellier, in Germany at 2 sites, Heidelberg and Tübingen, and then Naples, Italy, and then Lisbon, a center in Lisbon in Portugal. So I think that you can see from this slide that the CRF is not it's not a United States entity. It truly is a global entity. We are in talks right now with investigators in Israel. We are in talks with an investigator in Kenya. And we have had individuals as far away from at the International Choroideremia Research Network, which we'll hear more about. We've actually had on those phone calls at any one time, we'll have individuals from all 6 continents, just Antarctica is excluded. Just a few slides here on the recent scope of funding. You're going to see there's sort of 3 broad categories. First is a pathophysiology. This is exactly the molecular mechanisms as to what causes choroideremia. This morning we heard that the actual molecular interactions or lack thereof of what actually causes choroideremia or the deleterious effects of missing the REP1 protein. We still don't totally understand that molecular mechanism. And so understanding the basic pathophysiology we feel is very important for the development of therapeutics. And so we're looking at the function of the REP1 protein, organelle crosstalk, vascular biomarkers and how that might be affected in the absence of REP1, phagosome maturation, apoptosis in that is cell death in individuals with choroideremia, mitochondrial defects in individuals with a REP1 mutation, and the use of induced pluripotent stem cells. And as you heard in this morning's discussion, that really, in the past several years, the induced pluripotent stem cells has really taken off. So just in addition to the basic science, what about in the clinic and other therapeutics and treatment? We've funded research in the last 5 years on adaptive optics, DNA nanovectors, how to measure photoreceptor structure and function. This would be very important because as we're moving forward with additional trials, how do you determine, other than using an eye chart, how do you determine if somebody has had a positive or negative response to a therapeutic? And so understanding how to measure the photoreceptor structure and function could be very important to that. We're looking at ways of to enhance neuroprotection, induced autophagy, just mapping of the visual brain, meaning when the retina is activated, where does that go into the brain, and can we use those signals to identify whether or not a therapeutic is helpful or not? As I mentioned, we're going to look at female carriers, and so we're looking at the phenotypes of female carriers, meaning if a woman is diagnosed in her 20s, 30s, 40s, 50s, where is she along on the spectrum of her own eye health, and what can she anticipate? We're looking at transcript RNA read-through technologies as a way for a therapeutic, and then antisense oligonucleotide therapies as well. And then finally, just sort of a hodgepodge, we've got two brothers that have the same genetic defect, but they have totally different expressions. One is very symptomatic, one has almost no symptoms whatsoever. And so Dr. Ian McDonald in his lab is trying to figure out what the different, like why would two brothers express their genes very, very differently? And then as was mentioned today, and I've already mentioned a little bit, we're really looking strongly into animal model development so that we can test therapeutics and have other research vehicles and vectors. And we'll hear more about that in just a few minutes. So just a brief slide on how funding works through the CRF. We have what's called a rolling receival of grants. That is, most of the time an investigator comes up with an idea and they contact us, and then we use the same sort of grant form that the National Institutes of Health uses to grade and review their grants. Every once in a while, like for example, we'll do a call for proposals. So we will distribute to thousands of individuals on our mailing list, our email mailing list, and say, we're going to do the Wheelock Award. It's an award for this amount of money, and this is what we're looking for. And so that will happen on occasion. Once the grants come in, we review the grants with our scientific advisory board, and we are instituting the same sort of structure that the National Institutes of Health uses, and that is so these are PhDs whose background is in just scientific ophthalmologic research. We have one of those individuals do a primary thorough review. We have a secondary reviewer, and then everyone on the science advisory board can then weigh in with their thoughts on the project. Once the science advisory board has given their recommendations, we then look at the recommendations and the grants themselves with the research committee. Once the research committee has decided which grants should be funded, we then present our recommendations we give our recommendations for consideration to the board, and then they ultimately decide whether or not to fund these projects. And then we circle back to the grant submitter of whether or not their project was funded. And often, if it's not funded, I will meet with the investigator on the phone and and we will discuss about ways that we might be able to enhance or alter the project so that we can fund it. So in summary, we have an incredible personnel, not just on the research committee, but also on our scientific advisory board. They do a ton of work behind the scenes to make sure that the funds that we receive are used in a thoughtful and wise purpose. I hope I was able to show you that we really do try to take a very strategic approach to the grants that we fund. Over the past several years, we have increased the amount of grants that we've been able to fund, and this is all because of generous donors and fundraisers like yourselves. So thank you for all that you do. Again, I'm going to break off here for a few minutes. If you have a specific question or two about the research committee, the scientific advisory board, or how we do it, I'll take a few quick questions now. We will have another presentation, and then again I can take questions then as well. Again, that's an incorrect email. It's Jess Thompson. There's no dot. It's just Jess Thompson at curechm.org. I would love to hear your comments or your thoughts and ideas. And yes, sir, with the hand up. Yeah.
You mentioned that there's been a focus on research involving female carriers and the effects on them. Is that focused on treating the effects on them, or is there some thought that there might be a focus on keeping the progression from a path forward, stopping them from passing it on?
Thank you. So if I heard you correctly, and just so everyone can hear, so the question is, and please tell me if I'm not stating this correctly, but is the focus of the female carriers more on a therapeutic to stop or halt the progression of the female carrier? Is that so? So right now we're just trying to understand the female carrier. We don't even have basic data on the female carrier. So if a woman in her 30s, let's say, goes in and they say, often they say you have retinitis pigmentosa. Oh, oops, you actually have choroideremia. And at age, let's say 30, so they can't even tell her, based on what we see now, your likelihood of progression by age 40, 50, 60 is this. And some women will maintain their vision well into their 70s and 80s. Some will be almost blind by the time they're in their sixth decade. And so why does that happen? And so right now our efforts have been mostly trying to describe female carriers, meaning if they have this gene, that their likelihood of their disease progression is this, because even that basic research hasn't been done. The hope is though that whatever therapeutic that we use for affected individuals with choroideremia, those same therapeutics hopefully would also be useful in a female carrier. Any other quick, that's a great question. Any other quick questions before I scoot and introduce our next speaker?